I welcome you all for the course in cell culture technology. So, this course essentially is one of the very important tool for any kind of biological research, especially dealing with animal cell culture. We all know that for any kind of understanding of mechanism of biological phenomena or for screening different kind of drugs or understanding the effect of different parameters, much before we do a animal trial, we try to see the effects in dissociated cells in a culture dish. So, once we have a fair idea about how the dissociated cells are reacting to certain conditions, certain drugs or certain situation, we extrapolate our data for the whole animal studies. So, it involves a series of procedures where how we can isolate the animal cells, what are the different experimental animals which are being used, what are the basic protocols which starts from what kind of safety measures one has to take, what kind of cleanliness is needed, aseptic measures which has to be taken, how to set up a lab, how really to grow cells, how to develop cell lines, how to do genetic manipulation in them. So, this whole area falls under the different techniques, and most of the places are having good cell culture labs as a very basic prerequisite for any bioengineering, biology or bioprocess engineering department. But yet there are many places where people are not trained how really to set up a lab like that at a very small budget, because we all run through a budget constraints. Okay. So, here what I will do, starting from the very basic fundamentals, the historical perspective, how the cell culture has started during the last century. It started somewhere around 1900, very early 1900. Since then, different cell culture technologies, cell culture techniques, medium development, patterning of cells. Now, currently the whole pattern is about three-dimensional culture, which is used in tissue engineering and biomaterial based application for clinical material testing. How this whole thing has evolved through different time points and what are the technologies involved, and on certain occasions I will take a tour of the lab which will have the video which will give you an idea wherever you are sitting, could you set up a lab at a very minimum budget and yet you can produce really significant results. So, our goal will be to have a overall understanding of last more than 100 years, how this field has evolved, what are the different technological challenges, different technical know-hows, and how this field is progressing in supporting biomedical research all over the world. So, it will be in a way a virtual hands-on course which will expose you or train you to set up a lab for cell culture and all other allied procedures which are currently needed in all formats of modern biomedical research. So, thank you once again and I welcome you, and let us learn together this wonderful world of animal cells under a microscope and how that whole technology is helping us to get a better insight about who we are and how most of our cells function. Thank you.